Hello, welcome to Teacher Gia's channel. Thank you for clicking onto this video. Our topic for today will be the cell. I believe that all of us, if not most of us, have heard the word the cell, which is in science and biology. So at the end of the lesson, you should be able to 1. Define a cell. 2. Name the parts and functions of the microscope. 3. Understand all the rules in handling a microscope. And lastly, know how to use a microscope. So let us define a cell. A cell is a small compartment that holds all the biological equipment necessary to keep an organism alive. It is the basic functional unit of an organism. So that is a cell. Next is that organisms that are made up of single cells, such as amoeba, are called unicellular organisms, while those that are made up of many cells are called multicellular organisms. Most plants and animals are multicellular. Another thing is that a cell, being very tiny, cannot be seen using naked eyes and therefore require a powerful magnifying lens. More often than not, a microscope is always used to view the cells. So what is this microscope? We move to parts and functions of the microscope, and before that, let's talk about this light microscope first. It's commonly used in schools because it does not focus so much on the internal structure of the cell. It uses a beam of light to illuminate the specimen being viewed. Total magnification of the specimen viewed under a light microscope is given by magnification is equals to eyepiece lens magnification times objective lens magnification. So that is the formula for calculating total magnification of the specimen. So for example, if you are given the eyepiece lens magnification is times 10 and the objective lens magnification is times 8, then the total magnification will be 10 times 8, that will be 80, and you have to place that x or times in front of 80 so that it becomes correct. It is a must. Take note of that. So next we're going to talk about parts of a microscope, and as you can see, I have a figure here with all labeled parts, and besides that, you're also going to denote all the functions of each and every part. So let's begin. So the first part is the limb or the arm. As you can see, I'm pointing into it. So it supports the body tube and the stage. So that is its function. You can see the body tube and the stage that are being supported. Next, we're going to talk about the base. It provides firm and steady support to the microscope. The body tube. It holds the eyepiece lens and the revolving nose piece, so that is its function. The coarse adjustment knob. It raises and lowers the body tube for longer distances to bring the image into sharper focus. Next, we move to fine adjustment knob. It raises or lowers the body tube for shorter distances to bring the image into sharper focus, mostly used with the high power objective lens. Diaphragm, an aperture that regulates the amount of light passing through the condenser to illuminate the specimen. That is the function. Next is the eyepiece lens. It has a lens that contributes to the magnification of specimen under review. Another lens is objective lens. It brings the image into focus and magnifies it. Next is a mirror or illuminator. It reflects light through the condenser to the object on the stage so that the object can be viewed. A revolving nosepiece holds the objective lens in place and enables change from one objective lens to the other. Next is the condenser. It concentrates light on the object on stage. Then the stage is a flat platform where a specimen on the slide is placed. It has two clips to hold the slide into position. And those are the parts and functions of the light microscope. Next, we move to rules in handling microscope. Rule number 1 is clean hands. Ensure your hands are clean before touching the microscope to avoid transferring dirt or oils. The second one is carry with 2 hands. Hold the microscope with both hands, one on the arm and the other supporting the base to ensure stability. The third one is lowest power objective first. Always start with the lowest power objective lens before progressing to higher magnifications. The fourth one is use coarse adjustment carefully. When focusing, use the coarse adjustment knob only with low power objectives to avoid damaging slides. Next, switch off before changing slides. Turn off the microscope before changing slides to prevent potential damage or misalignment. The sixth one, keep stage clean. Wipe the stage with a lens paper after use to remove any residual oils or debris. Next, use lens paper. Clean lenses with lens paper, not fingers, to avoid leaving smudges or oils on the lenses. Next is store properly. After use, lower the stage, remove the slide and store the microscope with the low power objective in place. And the last two are be gentle. Handle the microscope gently and avoid banging or bumping it, as microscopes are delicate instruments. Last, follow laboratory guidelines. Adhere to any specific guidelines or protocols provided by your laboratory teacher or instructor. I hope that's clear. And on to our last topic, how to use the microscope. So the procedures are: prepare the microscope, place the microscope on a stable flat surface, ensure that it is clean and free of dust. Then you turn on the illuminator. If the microscope has a light source, turn it on to illuminate the specimen. Then after that, you adjust the brightness of the light source for optimal visibility. Then you place the slide, put the prepared microscope slide on the stage. The specimen should be facing up. Then select the objective lens. This is by bringing the lowest power objective lens, usually 4 times or times 4. Rotate the nosepiece to select the desired lens. Then use the close adjustment knob to bring the objective lens close to the slide without touching it. Then after that, you can now use the fine adjustment knob, which is used to refine the focus using the fine adjustment knob for clear image. The next one is you move to higher magnification if needed. If needed, switch to higher power objective lens, times 10, times 40, times 100 using the nosepiece. So the nosepiece is used in doing that. Always ensure to refocus as necessary. Refocus with the fine adjustment knob each time you switch to a higher magnification. Then examine the record. Observe the specimen carefully and record any observations or findings. And the last two are turn off the illuminator. Once you're done, turn off the light source. Then store the microscope. Lower the stage, remove the slide and store the microscope properly. Remember to follow any specific instructions provided by your teacher or laboratory guidelines for microscope usage. Never forget you're supposed to follow each and every instructions you're given by your teacher or a laboratory assistant. And we have come to the end of this topic. Thank you for clicking onto this video. I hope you've learned, and see you in the next video. Bye.